We have talked about briefly about focal adhesion kinase or FAK. I feel that we need to discuss it a little bit more because it plays a very important role in cellular adhesion. So let's look at some of the features of FAK. Signaling functions of integrins greatly depend on the cytoplasmic protein tyrosine kinase called focal adhesion kinase or FAK. FAK is one of the major tyrosine phosphorylated proteins found in focal adhesion. When integrins cluster at the site of cell matrix contact, FAK is recruited to focal adhesions. We have seen a role of FAK, how it is basically resulting in how the role it plays in linking up the integrin with the cytoskeleton. So it is a very important protein. It is recruited to focal adhesions by intracellular anchor proteins which bind to integrin beta subunit. The clustered FAK molecule cross-phosphorylate each other. We have seen that, for example, receptor tyrosine kinases, when they dimerize, the two of them come together, they cross-phosphorylate each other. Similarly, clustered FAK molecules also cross-phosphorylate each other on specific tyrosine residues, which results in creation of docking sites for cytoplasmic tyrosine, other cytoplasmic tyrosine kinases. One of them we talked about is, for example, SOC. FAK deficient fibroblasts adhere to fibronectin and form focal adhesions, but they form too many focal adhesions rather than too few. So we have seen that FAK plays a very important role in this linkage and linking integrins with the cytoskeleton. Also, the point here is that regulation of FAK is important because if it is completely deleted, so it can result in too many focal adhesions and prevent cellular prevent spreading of the cells or migration. FAK normally, which implies that FAK normally also helps disassemble the focal adhesions, and loss of adhesion is required for regular cellular migration. FAK can couple migratory signals to changes in cellular adhesion. Many cancers, for example, many cancer cells, for example, have elevated levels of FAK, which may help explain why they are often more motile than their counterparts. So that is one of the things that cancer cells can do, and are basically, if they have to metastasize, they have to basically disengage from the ECM in order to move to a different location. Here we have this experiment. You can see we have two photographs, one which is showing normal fibroblasts, the other the FAK deficient fibroblasts. Please notice that the normal fibroblasts, they have spread out. They're covering more area as compared to the FAK deficient fibroblasts. So these FAK deficient fibroblasts, they have been stained with vinculin antibodies to show, to reveal the location of focal adhesions. Here, similarly, these cells have also been stained with vinculin antibodies. So again, you can see these white dots. These are basically the focal adhesions which are found in these cells. Here you can see fewer on the left side. On this side, you can see fewer dots compared to these FAK deficient fibroblasts. Additionally, normal fibroblasts, they have spread out. This is after 2 hours after plating these cells, and this is also taken 2 hours after plating the cells. And you can clearly see that these cells have not. Basically, the point here is that FAK or focal adhesion kinase activity has to be regulated. Too much or too little can interrupt or disrupt normal cellular functions.